All right, today we're diving deep into the absolute bedrock of biology, amino acids. Seriously, these things are the fundamental building blocks of proteins, and if you want to understand life, you've got to understand them.
You know this quote from Francis Crick? Yep, one of the guys who figured out DNA just nails it. It really captures this beautiful, profound idea at the heart of all of life's complexity. It's like every living thing on Earth is written with the exact same tiny alphabet.
So that's the big question, right? How in the world can this staggering diversity of proteins, we're talking everything from the enzymes that power our cells to the antibodies that protect us, how can all of that be built from such a small universal toolkit? Well, let's find out. It really all boils down to this. Every single protein, no exceptions, is just a long chain, kind of like a string of beads. We call that chain a polypeptide, and each individual bead, each link in that chain, that's an amino acid. They are the letters in life's protein alphabet.
Okay, so to really wrap our heads around this, we need to look at the blueprint of an amino acid itself. Let's take one apart and see what they all have in common, and more importantly, what makes each one so special.
Every single one of these amino acids is built on the same basic framework. At the very center, you've got a carbon atom, which we call the alpha carbon, and attached to it are two really important functional groups, an amine group, which is basic, and a carboxyl group, which is acidic. And boom, that's literally where the name amino acid comes from. This shared structure is what allows them to link up with each other. So you see these first three parts, the alpha carbon, the amine group, the carboxyl group, that's the constant part, the backbone. But that fourth piece, the R group or side chain, that's where the magic happens. This is the only part that's different from one amino acid to the next, and it gives each one its unique chemical personality, which totally dictates how it's going to act inside a protein.
Now here's a really cool and kind of weird rule of life. Chirality. Most amino acids are chiral. What that means is they come in two mirror image forms that you just can't stack perfectly on top of each other. Think about your left and your right hand. We call them the L and D forms. And for reasons that are still one of the biggest mysteries in science, life on this planet decided almost exclusively to use the L form for building proteins.
Now, I bet you've probably heard that there are 20 amino acids. And for the most part, that's true. 20 of them are programmed by the standard genetic code. But the full story is a little bigger. The actual number of amino acids that we know get built directly into proteins is 22. So who are these two mystery guests? Meet selenocysteine and pyrrolysine. They're so interesting because they don't have their own unique code in our DNA. Instead, the cell's machinery performs this incredible trick where it sees what's supposed to be a stop signal, and under very specific circumstances, it ignores the stop sign and pops in one of these special amino acids instead. It's like a clever biochemical loophole that expands the alphabet of life. Okay, so now we have our full toolkit of 22 molecular building blocks. How do scientists even begin to organize them to figure out what they do and how they'll behave? Well, there are a couple of really useful ways to sort them. The first way to classify them is by nutrition, and this one has a direct impact on your life every single day. We've got the essential amino acids. Our bodies just can't make these, so we absolutely have to get them from food. And then there are the non-essential ones, which our cells are perfectly capable of making on their own. But, biology is never quite that black and white, is it? There's this third, fascinating category, the conditionally essential amino acids. Normally, your body can whip these up just fine, but when you're under major stress, like a serious illness or injury, your body's demand for them goes through the roof, and suddenly you can't make enough. They become essential, and you have to get them from your diet. Now, if you're a biochemist, the classification that really truly matters is all about the chemistry of that R group we keep talking about. Is it nonpolar and afraid of water? Is it acidic and carrying a negative charge? Or maybe basic and positive? Answering these questions is the key, because these properties are what will ultimately control how that long chain of amino acids folds up into a complex, working, three-dimensional machine. So, let's move away from these abstract categories and actually see how this works in the real world. To do that, we're going to spotlight a few of the key players and see how their unique structures give them some incredibly important jobs. First up is proline. I love this one. I call it the rule breaker because its structure is totally different from all the others. It does something really, really weird. Just look at that structure. Proline's side chain actually loops all the way back around and bonds to its own backbone, which forms this stiff, rigid ring.
This literally puts a kink in the polypeptide chain, making it super inflexible. It just can't fit into normal protein shapes like helices, so instead, it acts as a structure breaker, forcing sharp turns. This rigidity is absolutely vital for proteins like collagen, the stuff that gives our skin and tissues their strength.
Okay, next up, cysteine. Cysteine has a superpower, and it all comes down to one crucial little atom in its R group, sulfur. And that sulfur atom gives it a chemical ability that no other amino acid has. Cysteine's self-hydryl group is super reactive. So when two cysteine molecules in a protein chain get close to each other, their sulfur atoms can link up and form an incredibly strong bond called a disulfide bridge. You should think of these bridges as powerful molecular staples. They literally lock different parts of the protein together, which is a huge reason why many proteins have such a stable and robust 3D shape.
And finally, let's talk about valine. By itself, it's a pretty straightforward nonpolar amino acid. But its story, well, it's a powerful and pretty tragic example of just how critical the precise order of these amino acids really is.
So here's what happens. In hemoglobin, the protein that carries oxygen in your blood, the sixth amino acid in the chain is supposed to be a polar one called glutamic acid. But in people with sickle cell disease, a tiny genetic typo swaps it for the nonpolar valine. That one tiny change, swapping a water-loving amino acid for a water-fearing one, causes the hemoglobin molecules to stick together when oxygen levels are low. This forces the red blood cells into that sickled shape, which leads to all sorts of devastating health problems. It is a dramatic lesson that the correct sequence isn't just important, it can be a matter of life and death. Okay, so we have met some of the blocks, and we've seen just how important their individual properties are. So what's the last piece of the puzzle? How are they all actually linked together to build a protein in the first place?
The process is actually beautifully simple and just repeats itself. The carboxyl group of one amino acid reacts with the amino group of the next one in line. When they join, a molecule of water pops out, and a really strong, stable connection is formed. We call that the peptide bond. And then the process just repeats over and over and over, adding more amino acids to create that long polypeptide backbone.
And that right there brings us full circle. That's the solution to the big mystery we started with. It's an unbelievably elegant system. Just 22 different blocks linked together by one type of repeating bond. And from that simple setup, you get the foundation for the almost infinite variety and mind-boggling complexity of the entire world of proteins.
But this leaves us with one last really intriguing question. We know there are hundreds of other amino acids out there in nature that are not used to build proteins, things like ornithine and citrulline. So if they aren't the building blocks, what other critical secrets are they holding? What other jobs are they doing inside our cells? That's a story for another time. Thanks for listening.